So let's revisit our pie chart of mutations. Now, remember, and this is a little bit biased. In fact, I'm going to change this just a little bit, but there's going to be a bit of a swap here. So after natural selection and genetic drift, get to work on that pie chart. And remember, the pie chart was 95% neutral, 5% negative or so, maybe a little less than 5%, and 0.1%, just a tiny fraction, were positive. Now, what happens to our negative mutations? Well, natural selection is going to begin to work on them to remove them from the population. But remember, there are differing effects of natural selection on negative mutations. Negative mutations that are highly detrimental are removed very rapidly. And so we don't see those, and you might catch them very quickly if you're sampling large parts of a population or if you just accidentally randomly happen on it right after it occurs. But in the long term, those just go away because the individuals that carry them die and they don't get passed on in the population. However, in reality, because some negative mutations are protected by genetics, dominance recessive can play a role, or they don't play an impact until later in life after reproducing is done. So some negative mutations do stay in the population, but they're going to be a very small percentage. And remember that biologists in the past have been interested in the positive stuff. That's what Darwin was looking at, right? He's looking at all these good things, this endless diversity that makes hummingbirds different from other birds, that makes adaptive mutations and forms that just kind of capture the attention. And so for the first 200 years or so of evolutionary theory, and even before that, before we even knew about evolution, people were studying this 5%. And so in reality, it might be a better idea to make this chart and make a very slim pie piece for negative mutations because they are not removed immediately. They hang about at some low rate in the population for differing reasons. But remember that biologists historically were just interested in looking at, okay, what makes species A different from species B, what allows them to adapt and survive in the environment. And maybe they looked at a little bit of neutral evolution, at variety and diversity that was interesting but didn't really cause a fitness difference. But by and large, they missed the vast majority of neutrality. And even today, when people who are unaware of this feature of evolution, that most mutations are neutral, most of the diversity and variety that is in the genes of organisms doesn't make a difference one way or the other, right? That's a little jarring for some people and counterintuitive. Most of the diversity we see in populations and between species is completely neutral and doesn't make a difference one way or the other, whether the organism survives and thrives or dies off and doesn't pass on those mutations. And so most people think, oh, there's all that diversity, it's useless, it's neutral, we can't do anything with it. But that's not true. In fact, there are many very cool things you can do with neutral evolution that you can't do with things that are under positive selection. Now, by and large, you have to treat every positively selected mutation independently. It's going to have different features, different strengths, different outcomes than another one. And so we cannot treat positive mutations as one big group and study what's going to happen to them as a group because they're just too divergent. There's too much variety and diversity in those types of positive mutations. However, neutral mutations all have certain features in common, and taken as a whole, they can do some very powerful things for us. So let's look at 5 of the main things that neutral variety and diversity can allow us to do when we have big enough data sets. Okay? So the first thing, there are going to be parts of the genome that are under different selection pressures, and therefore they would exhibit different patterns. Right? So for instance, how are the protein coding parts of genes, or even the non-protein coding parts of genes, right? The introns, how are they different than the rest of the genome? And if we can define those differences over large numbers of genes, then maybe we could help us discover in general what forces are acting on genes that are outside or different than what's acting on the genome. And so the position, the most diversity occurring in these third positions in the codon, the reading frame, that's a very easy, obvious one, but there are lots of others. And so basically, we can treat neutrality as kind of like the null hypothesis. And then we look for those rare instances. Maybe it's only a very small part of the genome, but we look for those rare instances where it is violated. And if we see a violation of neutrality, then we have something cool and interesting we can look at. So it's probably worthwhile thinking about what would happen to these neutrally coding parts of the DNA. If we just had random mutations, not good or bad and no bias one way or the other, and if there's no natural selection acting on them, then we would be able to make some fairly straightforward predictions. We would expect to see all 4 base pairs at roughly equal frequencies, right? We'd expect to see, within a margin of error, about 25% of all the base pairs should be guanines, about 25% cytosines, and so on, right? It should be balanced. Now, remember, because we always have pairs, that we really can't treat guanines and cytosines differently when we're looking at genomes, because every time you've got a guanine, you've got a cytosine pair on the other side. And so we don't really treat 25, 25, 25, 25, but we do look at GC pairs, right? Because every time you have a guanine, you have a cytosine on the other side. And so we look at GC pairs versus AT pairs. And so we would expect to see a roughly 50-50 split. And if we didn't see that, that would violate our prediction. We would also expect to see no differences between different regions of the chromosome. So if I looked at the end of a chromosome and calculated all the statistics and patterns that I saw there, I would not expect to see anything significantly different than the middle of the chromosome or midway down the chromosome. Okay? So just be aware there are lots of really nice predictions, and when we violate those, we see some interesting patterns. So one of the biases we see are GC bias. And this can be on an entire genome. So for instance, there are some bacteria like R. sphaeroides that have a really high GC content. And again, under neutral theory, it should be close to 50-50, not significantly different. Notice E. coli is right around 50-50. A few areas of the genome have lower, a few have higher, but by and large and taken as a whole, it's right close to 50-50. This is P. falciparum, which is a eukaryotic organism. This is the microorganism that causes malaria. And notice it has a very, very low GC content bias. And so GC bias or GC content bias shows us there's some other force of natural selection, maybe acting on an entire genome level in this case. Okay, so we don't know exactly what that is just by seeing the bias, but now people are beginning to study and understand this. One explanation can be environment. If you are living in a very extreme environment, like a very salty environment or very hot or a very acidic environment, there tends to overall be a benefit for higher GC content because that makes your DNA more stable. Remember, the GC pairs, those nucleotide pairs together, in G and C pairings, they have 3 hydrogen bonds, but in A and T pairings, there are only 2. So GC makes the molecule, the DNA molecule, a little bit more stable, a little bit more thermodynamically unable to break apart to unzip, which is really good in some environments. And so there might be overall selection towards that. And that's just an overall that makes the genome more stable, but that's a beneficial thing. And that's maybe a little different form of natural selection than most people think of. Many people think of natural selection as a genetic mutation that has a direct impact on some very one particular trait. And I guess maybe, this is many, many mutations that all have an impact on an overall trait of having a higher GC content bias. Now, the other type of bias that we can see is codon usage bias. And again, if we have completely random mutations accruing, then when there is degeneracy at the DNA code, so let me give you an example, and I'll actually type it in here. And this is the only one I have memorized. CCC codes for proline. I know start codons too, but I figure why would I need to memorize them if we have little charts that show it to me? So CCC codes for proline. But so does CCG, and so does CCA, and so does CCT. At the DNA level, we could replace the T with a U if we're looking at the RNA level, okay? So, under neutral theory, if I looked at all the prolines in a certain gene, or maybe even all the prolines in every gene, I would expect to see roughly, because there are 4 different possibilities, and this third one can mutate randomly and then with no impact of natural selection acting on it, we would expect to see about 25% of each of these codons represented across the gene or across the genome. And so if there's a bias there for some reason, then there's some other natural selection force acting on. And there can be multiple explanations for codon usage bias. And realize these 2 concepts may not be completely separate from one another. If I'm in an organism with a high GC content bias, then I would expect to see CCC and CCG represented more frequently in the genome than a CCA or a CCT, because we're biasing towards having that cytosine or that guanine at that position. And so codon usage and GC bias can be connected. Now, there can also be regional patterns. We looked at overall genome patterns in these three organisms, but there can be regional patterns for these. An isochore is a region of a eukaryotic genome that has a significantly different GC content. So notice GC content is right a little bit, it averages a little less than 50%. There might be a little tiny, maybe significant fraction of overall GC content. But then regionally, we can see different areas where we might have a high GC content bias in some areas and a low that corresponds to those two little peaks right there, and a low GC content in another area. And so we call these isochores. And so what that means is there are selective pressures, and it might be for stability, but there might be some other force that we maybe need to find and investigate. So this might be just right about, not different from the average and where we'd expect to see under neutrality, but these are significantly different. And so by identifying those significantly different areas, we can begin to look at very cool evolutionary forces that before were just beyond our ability to estimate. So know the definition of isochore, and know that it can play a role and can be correlated with chorons, right? Codons. So if I'm in a very low GC content area, I might expect to then see a corresponding bias in the codons that are in that area that would have fewer G's and C's. Okay, and it's not huge in eukaryotes. This I think it was taken from a human, but it's enough that's significantly different, and so we need to investigate it. Okay, the third thing that we can do with neutral theory. Under neutral theory, we would expect the diversity within a population, so diversity measured at the species level, to be correlated at diversity between species. So for instance, if Drosophila melanogaster and Drosophila similans are very, very diverse species, then we would expect these to be very long branches, that there'd be lots and lots of differences and changes along this branch. Whereas if we look at a gene and say, oh, Drosophila melanogaster, there's almost no diversity at the species level for that gene, and for simulans, there's no diversity at the species level for that gene, we'd expect there to be very little divergence when we compared the two genes across species. Okay. Now, in the paper that you're reading, the Ney 2011 paper, it talks about the McDonald-Kreitman test. And the McDonald-Kreitman test looks for violations of this underlying assumption. If I see lots of diversity in Drosophila melanogaster and very little diversity in simulans for a gene, or lots of diversity here, but very little differences, that's very unlikely. But if we did see it, that would tell us where natural selection. So let's look at a more likely example. Let's say Drosophila melanogaster for maybe a sensory gene. And we see tons and tons of diversity across that sensory gene in Drosophila melanogaster. But when I look at that diversity in simulans, it's very, very low, and this branch is very short, and maybe even that branch is very short, but then suddenly within Drosophila we have tons and tons of changes. Well, that could mean that then suddenly in the species, that gene has been under strong, what we would call positive selection. So positive selection occurs when we have significant forces that change a gene to take on a brand new function. Purifying selection, sometimes called negative selection, but that term is not used as commonly because it can be misconstrued. But purifying selection is the opposite of positive selection. And purifying selection occurs when we have very little diversity in a gene and especially very little functional diversity. So for instance, if I look at a gene that helps to plays a role in cellular respiration, a really critical gene, I'm very likely, if I look from person A to person B to person C, or fruit fly A to B to C, whichever population you're looking at, I'm very unlikely to see any differences, any functional differences in that gene. So that would show that it's been under very, very strong purifying selection. Now, in a later discussion, we're going to talk about this non-synonymous to synonymous mutation rate and how we use it to identify areas. But just review the McDonald-Kreitman, know generally how it works. And then for now, there's some of it in the reading, but for now you can kind of just put a pin and say, oh, we'll learn about the DN-DS ratio, the synonymous to non-synonymous ratio. We'll learn about that at a later date. Okay? One other thing, again, if we start with this estimation of neutrality, then when we find things like a haplotype block, good vocabulary word, right? And that has to do with, so let's actually replace isochore here with haplotype block, since we already did isochore. Isochores can play a role in this, but haplotype block's a little bit more of a relevant term here. Okay. So, when a brand new mutation occurs and it's really, really great, right? Really beneficial, it goes to fixation very rapidly. So notice over just a few generations, we have one individual, soon every individual carries that allele. And the white allele disappears from the population because of the strong selection for this allele represented by the dark square. But recognize also that not only is that thing going to be selected for, but genes that are right next to it are going to come along for the ride. And this is called genetic hitchhiking. So notice not only did we fix the beneficial allele, but we fixed the other two alleles that just by chance, the dark triangle and this kind of light colored circle, we fixed those alleles along with it. This is genetic hitchhiking. And so if we see a large region of a chromosome with very, very little allelic diversity across the genes in that region, that's a strong sign that one of those genes was selected for positively. And it doesn't have to be 100% like in this example. But if we see a much higher correlation than we should based on the function of that allele, it means it's come along for the ride. It's genetically hitchhiked. And so again, our assumption of neutrality is that there should be no correlation between allelic diversity at one site and allelic diversity at another site right next to it. But if there is, boom, that's a strong sign of natural selection for one of those alleles. Okay. Now, this last one I'm just going to mention because we have an entire discussion in our next unit about it. Okay. So the molecular clock is perhaps the most important and powerful, well, it's a very important and powerful prediction of neutral theory. Okay. Areas of the genome that are evolving truly neutrally could be used to help estimate divergence times. That is what molecular clock is, and we'll look at how it works in our next discussion. But for now, know that definition.